approximately 50% of all antibodies that are sold appear not to work. And if you calculate the amount of money that's being wasted, it's about $800 million a year worldwide on antibodies that don't work.
The whole field is suffering from poor specificity of antibodies. Many antibodies react to multiple targets. There is sort of non-specific binding to Fc receptors on cells in your that you're trying to stain. We spend lots of time, months, almost years, to establish working panels that we can trust.
It's a limitation. So we still use those antibodies because it's the best we have, but it's not the best.
As a human immunologist, a lot of the work we do involves phenotyping of cells by flow cytometry. It's crucially important for us that we have high quality reagents to minimize variation between experiments.
One of the main issues with fluorescence cytometry is the background fluorescence that is different from cell type to cell type. It's not only how good is my positive signal, but how low can be my negative signal.
Well, I published a paper in Nature in 2015, I think it was now, with Andreas Pruchthun and about 150 co-authors. And in that, what we said was that it was time to move on from monoclonal and polyclonal antibodies, and that all antibodies in the future should be recombinant antibodies.
Recombinant antibody technologies allows for more predictable production, I guess, of the antibodies, and also by allowing to engineer the antibodies for reduced binding to Fc receptors, for example, you can reduce background noise and non-specific binding, and that's what we have seen in initial testing.
Miltony has in recent years brought a whole catalog full of so-called REA or REAfinity antibodies. All of those are produced in culture. All of them come as an IgG1 subtype. All of them have a mutated Fc receptor binding site, and all of them are basically very reproducible and high in quality.
The REA antibody we have used, we have seen that there is a huge advantage in terms of the background signal. And I think this is one of the most obvious thing that we observe. And this independently of the fluorochrome that was tagged to the antibody. This is where technology brings value to science.
I think it's very much a matter of time. It will be as unacceptable to use a polyclonal antibody in 10 years as it is to use a gene that you haven't sequenced.